Welcome to your own channel Vedantu NEET English students. So before we start any topic for today, quickly let me know in the chat if my voice is audible to all of you, sound check, and the presentation behind me is visible to every single one of you. Quickly let me know in the chat if I'm audible and visible to every single one of you. Yes? Quickly let me know in the chat. I believe I'm audible and visible. Amazing. Now, today is going to be the part 2 of your RNA batch, second lecture from my side, that is respiration in plants. Previous class we did your photosynthesis in higher plants, done and dusted completely. We did a lot of concepts as well as question solving, right? Now we'll continue the same pattern in today's class also. We'll be solving concepts after concepts. After every single concept, I have many questions for you. In fact, I have more than 25 questions today, more than 25 questions, and all of them are last 5-year NEET PYQ, right? All of them are last 5-year NEET PYQ, and you will love them. And you will realize that every single time you understand a concept, immediately you're able to solve a NEET PYQ. So that will give you confidence that yes, whatever I'm learning in this class is actually beneficial to you. Right? So before we start the class, quickly everyone smash that like button, all of you. Quickly, Hulk smash the like button right now. And if you're new to the channel, consider subscribing to the channel. Okay. Now, before we go to the chapter, what is the weightage? Right? What is the weightage of this chapter which everyone talks about? See, first of all, tell me in the chat, how many of you think this particular chapter is a major backlog in your NEET journey? Tell me in the chat. Be honest. Be honest. This chapter is very technical chapter because it demands your concentration as well as your skills to understand the concept. So be honest and tell me, is this chapter moderately or difficult for you? If yes, I'm going to make this chapter very easy for you. By the end of today's class, you will be able to tell me, yes sir, I have completely conquered this chapter with confidence. Okay. Now, what is the weightage of this chapter? How many questions you can expect from this particular chapter right here? What could be the weightage of this chapter? Students, to be honest, if you look at your NEET 2024 paper, there were 5 questions from this chapter. That's right, 5 questions. So, you can, so this year it was around 20 marks. This year it was easily 20 marks. But the usual range for this particular chapter is from 16 marks to 20 marks. Students, that is the range. That is the range today. 16 marks to 20 marks. And if you want these 16 to 20 marks, if you really want to, make them yours, keep it in your pocket and go home today. Watch the lecture till very end, because every single concept from this chapter, throughout the chapter, you can expect some questions. Throughout the chapter, you can expect questions from here. Okay. So quickly everyone, green hearts in the chat to show your love towards the botany. And we will start today's class, that is respiration plants. Beautiful chapter, very easy chapter. Okay. I will make sure you understand every concept and not by heart it. I'll make sure of that. You understand every concept and not by heart any of it at all. Okay, now starting off with what is respiration? Wait a minute. Before we go to respiration, which is a chemical process, what is the difference between respiration and breathing? What is the difference between respiration plus breathing? So you should know the simple difference first of all. Breathing, inhale, exhale. Breathing is a much more physical process. It is a physical process, while your respiration is said to be a chemical process. It is a chemical process. That's right. It is a chemical process. That is respiration. Now, a simple question arises. What is the difference between respiration as well as photosynthesis? What is the difference between respiration and photosynthesis? Very simple question, right? What is the difference between respiration and photosynthesis? Very simple. You should know it very clearly by now. Photosynthesis is a process in which plants prepare their own food, that is inorganic, and light energy is converted to chemical energy as well as inorganic substance is converted to organic substances. Organic substance. The end product of your photosynthesis is nothing but glucose. Is nothing but glucose. The end product of your photosynthesis is nothing but glucose, right? It is glucose. Glucose. Now what happens in respiration then? What happens in respiration? See, in photosynthesis, plants prepared glucose. Plants prepared glucose. All the energy is trapped inside the glucose. In respiration, you take the energy out of the glucose now. Are you understanding? This is the first part. What do? In first part, in photosynthesis, plants are taking inorganic substance or light energy and converting into glucose. But in the second part, all the energy which is inside the glucose is extracted out and it is stored in the form of ATP. That is respiration. The second part. Now students, if you want to understand it better, let me give you a quick example of a cell. This is a plant cell. This is a cell. Not plant cell, this is an animal cell. Okay, animal cell. Now what is happening? Oxygen is entering inside. Yes sir, oxygen is entering inside. The food is entering inside. Now once the oxygen comes in contact with the food gets oxidized. And when the food gets oxidized, energy is released. Yes, energy is released. And that energy is stored in the form of adenosine triphosphate. That is nothing but your entire respiration. That is why is it in CO2? It's supposed to be O2. Oxygen. It's oxygen. Right? Oxygen is acting on the food. The food is getting oxidized, and once the food gets oxidized, it produces energy, and the energy is stored in the form of ATP. Any doubt here? Tell me in the chat quickly. Any doubt here? Quickly tell me in the chat. This is your entire gist of respiration. That is, the first part is called a process in which plants are producing glucose. The food right here is nothing but your glucose. Now once onto the glucose, oxygen is acted. Now once oxygen is acting on the glucose, the food is getting oxidized, energy is released, the energy is stored in the form of ATP. Adenosine triphosphate. That is the entire gist of your chapter. Now, what is respiratory substrates? Or respiratory substrates? Yes. Now, respiratory substrates are the substances. They are the substances on which biochemical reaction take place. For example, I mentioned food here, right? It is the same food which we call respiratory substrate. Now this respiratory substrates can be carbohydrates. It can be fats. It can be proteins, or it can be organic acids, right? Or it can be organic acids. These are your respiratory substrates. These are different types of respiratory substrates. Now, what about minerals? What about minerals? What about hormones. What about water? Are they also respiratory substrates? Can we utilize minerals, hormones and water for respiratory substrates, and also vitamins? Can we use them as respiratory substrates? Can we use them? Can we use the following substances for respiration to extract the energy out of it? No. No. We cannot use the following substances for respiration. We can only use carbohydrates, fats, proteins and organic acids for respiration. So someone asked me, sir, students, is my voice not audible to all of you? Is my voice not audible to all of you? Tell me in the chat quickly. So why plants take oxygen? Because I'll tell you, the main role of oxygen is actually towards the very end. The main role of oxygen is main role of oxygen is at the very end of your electron transport chain. Is it audible? Students, is it audible or is there any difficulties? Let me know the chat quickly right now. Yes, this is complete NCERT breakdown itself. If you read NCERT, see, after reading my notes, if you just read NCERT, it's going to be like a butter. Is it proper now, students? Check, sound check. Awesome. Arun, Anu, Janvi, Jai. It's audible, right? Perfect. Now, these are called as your respiratory substrates. Beautiful. Now, what exactly is ATP then? What exactly is ATP? Now, and how is ATP formed? What is the use of ATP? That's what we learned now. ATP is called as your adenosine triphosphate. Do you see this? Adenosine triphosphate. This is your ATP. But how is the ATP formed? How is the ATP formed at all? That is very important. How is ATP formed? So students, if you want to know how ATP is formed, you need to understand the entire process of your glycolysis, Krebs cycle, everything we learned today. But right now you should know that the basic requirement for formation of ATP, the most basic requirement for formation of ATP is going to be ADP plus phosphate. ADP plus phosphate. So this phosphate is added to here and we get 1, 2, 3 adenosine triphosphate. And students, now I told you this phosphate is added here. Can we use some glue? Can we use Fevicol? Tell me, can we use Fevicol to add it? Is my voice too low, students? Is my voice still low or is it high or low? Tell me quickly in the chat. If it is low, I have to call someone, that's why. Is my voice low? Is it breaking still? 1 second, students. One second, students, let me get corrected. Now, by then you tell me, can we use Fevicol to add this phosphate here? Can we use Fevicol? Yes or no? Can we use the Fevicol there? Tell me. If yes, tell yes. Is it breaking still? Sometimes it's breaking, huh? 1 second. This mic might be loose little. No. So obviously you cannot use Fevicol. Right? So obviously there should be some bond here, right? There should be some bond formation should happen here. Bond formation should happen there. Yes. Students, listen to me very carefully. If you want to add this phosphate to this phosphate, the bond formation is happening, right? All the energy is stored in that bond. So tell me, if you want to take energy again back from your ATP, how do you take? If you see, I told you all the energy is stored in this bond right here. Now, and we finally get the ATP. ATP is here. Now, if you want to remove the energy from it, what do? You break the bond. Obviously, you break the bond. So every single time you break the bond again, energy is released and we end up getting ADP. We end up getting ADP. So we have ADP plus phosphate ATP. From ATP again you break the bond. What happens? The energy is released and again you end up getting ADP. Clear all of you? Breaking the bond, you break the bond, you get the energy. Now how do you define ATP? All of us know ATP is called as your energy currency of the cell. All of you know that. Now why? Why do we call it as energy currency of the cell? Very simple answer. Now imagine your parents are working. Your parents are working and when they work, they get money into their bank account. Yes, they get money into their bank account. Right? The money into their bank account. Now once you go to shopping, you want to buy new dress, you tell your dad or mom, Mom, can you buy me that dress? What will they do? They will either take the cash or they'll take the credit card, debit card, or they can use UPI. And all the money which is present in the bank account is transferred to do some work, right? To do some activity or to get something. Similarly, students, inside your body, all the energy is stored in the form of ATP. All the energy is stored in the form of ATP. Like how money is stored in the bank, right? How money is stored inside the bank, inside your body, energy is stored in the form of ATP. And now every single time there's a requirement. What happens? Every single time there's a requirement, ATP bond is broken and energy is given out. Just like when there was a requirement for you to buy the dress. From the bank account, money was taken out. From the ATP, energy is removed out. Energy is removed out. Clear? So students, that is very easy to understand. But what is the meaning of this NADH and FADH? NADH as well as your FADH2. This NADH2 and FADH2 are like check. What do you do for check? You take the check and you give it in bank. You take the check and you give it in the bank and the bank will give you money. What is money? Money is ATP. So similarly inside the cell, NADH is given and we get the ATP back. That is why from NADH we can obtain the ATP. Beautiful, right? That is your ATP synthesis and NADH. Now, what is the difference between autotrophs as well as heterotrophs? So all of you know the basic difference. All of you will tell me, sir, autotrophs are the organisms which prepare their own food. Heterotrophs are the organisms which take food from the outside. But can we represent it with the help of a diagram? Yes, we are biologists, right? We are biologists. So we will illustrate with the help of an example. Say this, simple difference. Right? Here we have a plant. Imagine we have a tree here. Okay? We have a nice tree. Here. Now imagine here we have one, let's say, one human is there. Okay, Okay, let's take whose name should I take? Sir, is NADH used to break the bond? No. NADH is a separate molecule. NADH will produce ATP. Now, within the ATP, when the bond is broken, then energy is given. Your NADH will never give you directly energy. Check, with the help of check, you cannot do things directly. The check will always go to the bank. From the bank, money will come. That is the process, right? Check will go to the bank. From the bank, money will come. From NADH or NADH and FADH, you will get ATP. Then from ATP, you will get the energy. Remember that. Okay, let's take Sujit. We have a friend called as Sujit. Okay. Sujit. Now, what happens in the case of plant? Here. Okay, let's take this plant. Let's zoom in into the cell. Right now. And let's zoom in into one of the cells here. Let's zoom in. What exactly is happening inside the cell? What is exactly happening inside? Okay. Now, in the case of your heterotrophs, oxygen is entering inside the cell. This Sujit right here is making a sandwich. Even though his hands might be broken, he'll prepare a sandwich somehow. He's eating the sandwich. So the food is also entering inside. Food is also entering inside. The food is getting oxidized. The food is getting oxidized, and then energy is released. And then energy is released. Okay, energy is released. But in the case of plants, students, in the case of plants, oxygen is entering inside. Are we getting food from the outside? No. Why? Because we know, students, from the previous chapter, photosynthesis in higher plants, that plants prepare their own food. Yes. So inside the plant, there is something called as light reaction and dark reaction. In dark reaction, there is production of glucose. Yes. Food, which is glucose, which is produced inside the plant. Yes, inside the plant. We do not have to take anything from outside. Yes. Then it is getting oxidized. It's getting oxidized, as well as energy is released. Energy is released, it is trapped in the form of ATP. Energy is trapped in the form of ATP. Did all of you understand? Did all of you understand how it is the difference between autotrophs and heterotrophs? In the case of heterotrophs, the food is taken from external source. We cannot prepare our own food within a cell. Within a cell, we cannot. We take it from the outside. But in the case of plant, the food is prepared inside the cell. That's why they're called as autotrophs. That's why they're called as what? Autotrophs. Beautiful. So you don't have to buy out anything. It's going to be very easy. This chapter is very, very easy. Very easy chapter, students. Very easy. Especially Krebs cycle, very easy to understand. But the questions will be a little difficult. But I will make sure you solve all the questions also. Okay. Now, what is different type of respiration? One is called as aerobic respiration. Other one is called as anaerobic respiration. Aerobic as well as anaerobic respiration. In the case of your aerobic respiration, there is presence of oxygen. Presence of oxygen. Oxygen is present. In the case of anaerobic respiration, oxygen is absent. Oxygen is absent. Your aerobic respiration takes place in your cytoplasm, as well as your mitochondria, But in the case of your anaerobic respiration will only happen in the cytoplasm. It will only happen in the cytoplasm, does not happen in mitochondria. Very simple difference. Very, very simple difference. Very simple difference. Okay. Now, under your anaerobic respiration, we have two types. Okay. Under your anaerobic respiration, we have something called as fermentation. One is called as your alcoholic fermentation. Other one is called as lactic acid fermentation. Alcoholic fermentation happens in the case of your yeast, which is used to making alcohol. The other one is called as lactic acid fermentation, which happens in your lactic acid bacteria as well as your muscle cells. On the sports day, remember, on the sports day you ran 100 meters, you ran 200 meters. Next day you have all muscles are hurting. Lactic acid. Okay. So muscle cells as well as lactic acid bacteria as well as bacteria. Clear? So someone asking me, sir, isn't mitochondria in cytoplasm? Yes, it is in the cytoplasm. But some reactions are happening exclusively in cytoplasm and some reactions are happening only in the case of your mitochondria. I'll give you a quick example. The quick example is that in the case of your aerobic respiration, glycolysis is happening in the cytoplasm. Okay. Glycolysis is happening in cytoplasm. But what about link reaction? What about your Krebs cycle? Your Krebs cycle link reaction takes place in the mitochondrial matrix. Link reaction as well as your don't worry, we will get to this one by one. Link reaction as well as your Krebs cycle, citric acid cycle, both of them takes place in the matrix of mitochondria. Clear? It's going to be very easy to understand all these concepts. There's no difficulty at all. Very easy chapter to understand. Very easy chapter. Okay. Now, this is your entire aerobic respiration. See, under anaerobic respiration, you see, under anaerobic respiration, we have alcoholic fermentation and lactic acid fermentation. Students, one more thing you need to remember is your anaerobic respiration is always incomplete. It is always incomplete reaction. That is, incomplete oxidation is happening. Incomplete oxidation. Incomplete oxidation. But in the case of your aerobic respiration, there is complete oxidation or complete oxidation. Now, what is this complete oxidation? What is this reduction? All that we will learn. All that we will learn in today's class. Don't worry. All that we will learn. Okay. Okay, students. Now, this is your complete aerobic respiration. Glucose plus oxygen, energy is released, carbon dioxide plus water. This is a simple reaction which you've been learning from class 6, 7th, 8th. See here, glucose plus oxygen, energy is released. Where did this glucose come from? This glucose, in the case of your autotrophs, it's prepared inside the cell with the help of dark reaction. But the same glucose in the case of heterotrophs is taken from the outside. It is taken from the external source. See, now you should also know that aerobic respiration happens in multiple steps. It happens in multiple steps. That is, best example is glycolysis. Tell me in the chat quickly, how many steps are there in glycolysis? In the entire glycolysis, which is a multi-step process, there are totally 10 steps. 10 steps in glycolysis. In your glycolysis. 10 step in the case of your glycolysis. Clear? Now, where do plants get the respiratory substrate from? All of us know the respiratory substrates such as your glucose, carbohydrates, sucrose. Where do they get it from? Are they eating something else? No. All of you know plants get the respiratory substrate from themselves. That is photosynthesis. With the help of photosynthesis. That is, glucose is made in your dark reaction, and the same glucose is used by the plants for respiration. Now the next question is, do plants breathe? Do plants breathe? Simple question. Do you think plants are in the night plants, do you think they are secretly going and they are breathing something? They are breathing the air secretly in the night? No. So the answer is very simple. If you look at the actual answer, do plants breathe? No. Do plants breathe? No. But do they require oxygen? But do they require oxygen? The answer is very simple. The answer is yes. The answer is yes. They require oxygen. Plants require oxygen. So plants do not breathe technically. They diffuse in oxygen. They diffuse in the oxygen. Now, how do you think they breathe? How do you think they take in oxygen? For example, humans are breathing in through their noses. How do you think the plants are able to get the oxygen inside them? The answer is very simple. Let me give you a small clue. Transpiration loss. From where does the transpiration loss happens? Through stomata. Obviously, with the help of stomata, the same stomata which gaseous exchange happens, plants are able to take in oxygen. Plants are able to take in the oxygen. Right? Plants are able to take in the oxygen. Now, what about in the case of stem? What about in the case of stem? How does the oxygen enter through the stem? Thick woody stem. For example, students, thick woody stem. If this is your thick woody stem, on this thick woody stem, students, there are small minute gaps. There are small minute gaps on this thick woody stem. Small minute gaps. These small minute gaps are called as lenticels. They're called as what? Lenticels. So with the help of lenticels, plants are able to make sure that oxygen is entering inside. Okay. Now the next question is, why plants do not require special organs? For example, plants require food. Tell me, do plants require energy? Tell me in the chat. Do plants require energy? You'll be like, yes sir, plants require energy. Every organism requires energy. Now, inside the plants, obviously, all the food is prepared where? All the food is prepared in the leaves, sometimes in the stem. So, all the food is transported and all the water is also transported throughout the plant body with the help of xylem and phloem. The food which is prepared in the leaves goes to the root through phloem. Okay, this transportation is happening properly. But what about respiration? What about exchange of gases? What about transportation of gases? Do they have respiratory system? No. Plants do not have any type of respiratory system. No, not all present. Then how? How does the oxygen get transported throughout their body? The answer is very simple, students. Answer is very simple. The first answer is that plants has very less requirement. First of all, let me listen to me very carefully. Each part of the plant is self-sufficient. They can take care of themselves, every single plant. And also, tell me, what is the byproduct? What is the byproduct of photosynthesis? What is the byproduct of photosynthesis? Obviously, you will tell me, sir, one of the byproduct of photosynthesis is oxygen. It is oxygen. We tell, right? Plants take in CO2 and plants give out oxygen. So listen to me very carefully, students. Plants always, they don't make sure that some amount of oxygen is retained inside. Some amount of oxygen is retained inside during photosynthesis. So some of this byproduct is used by the plants. It is used by the plants for respiration. Clear? Crazy, right? Crazy, right? So some amount, remember, in photosynthesis that oxygen byproduct is there, right? The same byproduct is utilized by the plants. Utilized by plants. And also if you know, this is your stem. Again, this is your plant stem. This is your plant stem. Tell me, inside in the middle, everything is dead, right? In middle of the stem, obviously it will be dead. It's obviously dead in the middle. It's dead in the middle. So you should know this concept very well, that the living part of the stem, the living tissues are usually on the surface. All the mesophyll cells, all the parenchyma cells are only on the surface. Yes, are only on the surface. So all the oxygen which is entering inside, can easily be sent to every part. The oxygen which is entering on the surface can be easily sent to every single corner with the help of simple diffusion. With the help of simple diffusion, the oxygen can be transported everywhere. Okay. So plants do not require a special organ, because every single part can take care of itself. They're like, no, I will take care of me, you will take care of you. And also they have very less energy requirement. They have very less energy requirement. Very less energy requirement. Clear? Very less energy requirement. So plants do not require any special mechanism. Okay. Now, that was just the introduction to the chapter. It is very important. If you want to understand every single aspect of your glycolysis, Krebs cycle, as well as your electron transport chain, you need to understand the basics which you learned right now. So quickly tell me in the chat if there is any doubt regarding your first part of the class. I believe there should be no doubt. Everything was very simple. In a much more simple and intricate manner, we learned the first part of the chapter. What is autotrophs, heterotrophs with the help of a diagram. What does plants require? Do they breathe? No. Require oxygen? Yes. Okay. Now. Let's understand the first step. the first step in your respiration. Now, what is the first step in which glucose is undergoing? What is the first step in which glucose is undergoing? Can anyone tell me what is the first step in glycolysis? The first stepping stone in the entire, web of web of your respiration? Obviously, the first step is always your glycolysis. The first step is going to be your glycolysis, right? First step is going to be glycolysis. Now, let me tell you a few points about glycolysis and where does it happen, how does it happen, everything. But let me tell you that I will not be giving you any trick. No trick. There's no trick here. Because I'll tell you why. I'll tell you why there's no trick. Because the questions on glycolysis, they will never in the examination, they're going to ask you the entire flow of glycolysis. No. But they might ask you from which step to which step what is happening. They'll ask you. So if you really want to understand glycolysis, you need to go into the every single step, detailed diagram and structure. So that is what we'll be doing right now. We will truly understand glycolysis and not by heart glycolysis from now onwards. Okay, students, let's understand the glycolysis and not by heart it, shall we? We will understand it truly, that what happens in every step. What is reduction? What is oxidation? Okay. Now, students, first of all, we need to understand the location. The location of glycolysis. What is the location and where does it happen? The first thing we need to understand here is going to be the location. What is the location of glycolysis and where does it happen? Both in aerobic, anaerobic, prokaryotes, eukaryotes, in every single place, the location is cytoplasm. It is going to be cytoplasm. Location cytoplasm. Now, what about the number of steps? There are in total of 10 steps here. In total of 10 steps which we'll be studying today. 10 steps. Very easy to understand. Now, in the case of plants, this is very important. Since we are studying botany, in the case of plants, before glycolysis, there's one more step. In the case of plants, before glycolysis, there's one more step. That is, sucrose is converted to glucose plus fructose. And later on, this fructose is again converted to glucose. That is called as isomerization is happening. So remember this part very well. In the case of plants, sucrose is converted to glucose and fructose with the help of enzyme invertase. Enzyme invertase. Okay, that one thing you need to remember before we start the glycolysis, before we start the entire steps of your glycolysis. Okay. Now, before we start, there's one more concept which you need to be familiar with. What is the meaning of oxidation? What is the meaning of reduction? What is oxidation, reduction? Because throughout this chapter, on every single step, we'll be discussing what is oxidation and what is reduction, and I will not be repeating it again there. Learn right now. Focus 100%. What is oxidation? Oxidation is nothing but a loss. Oxidation is a loss of electrons or loss of proton. It's going to be loss of electron by a molecule, also loss of hydrogen. It is loss of hydrogen. Oxidation means loss. I'll write it for you once again. Oxidation is going to be loss. Oxidation is going to be your loss of electrons. Loss of electron and hydrogen, as well as your hydrogen. Now, what is reduction, students? What is reduction? Reduction is going to be your gain. Reduction is going to be gain of electrons and proton or hydrogen. Please remember this point right here. The entire, if you really want to master this chapter, students, if you want to master this chapter, this is the basics. This is the basics which you need to follow. This is the basics. If you know this, the entire chapter is going to be very, very easy. Entire chapter is going to be very easy. What is oxidation and what is reduction? Everyone quickly type this in the chat. All of you quickly type this in the chat right now. Savita, Ravindrani, Sebastian, yes, Pratibha, Divya, all of you. Oxidation is loss of electrons or hydrogen, this should be or, not on, or. Reduction is gain of electrons or hydrogen. Simple. Now, what is the other name? So, your entire glycolysis can also be called as EMP pathway. It can be called as EMP pathway. Now, why is it called as EMP pathway? Because it is based on the scientist who gave the term. Scientists who discovered glycolysis. There are three scientists. That is going to be your Emden, Merhoff and Parnas. Emden, Merhoff and Parnas are the three scientists who gave glycolysis. The first letter of the last name is used for EMP pathway. EMP pathway. Okay. Now, the entire glycolysis can be divided into two steps. Two broad categories. The first one is called as your preparatory stage, other one is called as your payoff stage. Think of it as a NEET journey. In your NEET journey, you are right now in preparatory stage. You're preparing, you're working hard, you're investing time. You're investing time and energy and money. Once you become a doctor, it is the payoff stage when you start earning respect. When you start earning money. Understand? That is called as payoff stage. Similarly, in your glycolysis, there are two steps. One is called as preparatory stage where you're preparing, you're investing. In payoff stage, investment is fruitful. You're getting the energy now. Okay, utilization is in the energy. Energy is being produced here. Now, what happens in your preparatory stage, students? I believe the entire glycolysis can be seen here for all of you. This is a snip of your NCERT to make sure we are on the right track and we can track every single step. Right? We can track every single step right now. This is going to be your entire glycolysis. There is no way you can by heart this. By hearting is useless. Understand every single step. Understand it. For example, the first step in your glycolysis is called as your phosphorylation. The first step is called as your phosphorylation. What is happening in your phosphorylation? In the first step of your glycolysis, your glucose is converted to glucose 6-phosphate. Yes, glucose 6-phosphate. If you by heart it, you will not understand because the question will come from here. The question will come from here. So tell me in the chat, what is the first step in your glycolysis? The first step in your glycolysis is phosphorylation. Phosphorylation. Okay, phosphorylation. Now, let's understand the structure right now. This is your glucose. Glucose is a 6-carbon molecule. Yes, 1, 2, 3, 4, 5, 6. To this 6-carbon molecule, we are adding a phosphate. And where is this phosphate coming from? The phosphate is coming from ATP, adenosine triphosphate. See here, ATP molecule. To this glucose, we are adding a ATP. Not exactly entire ATP. Can you see this? This bond is broken. This bond is broken right here. And this phosphate, you see the third phosphate. This phosphate is attached. This phosphate is attached to the 1, 2, 3, 4, 5, 6, 6th position. That is why glucose gets converted to glucose 6-phosphate because one phosphate is attached to the 6th position of your glucose in the presence of enzyme hexokinase. Very important. The enzyme name is very important. 6th position. So see this. So glucose to glucose 6-phosphate. 6th position, one phosphate is attached now. Now if the phosphate is attached to there, this is going to become ADP. Obviously adenosine diphosphate because one phosphate is missing right now. Now the second step, students. The second step is called as isomerization. Now let's keep a track of every single step. First step is done. Glucose to glucose 6-phosphate is done. 1 ATP is consumed here. Minus 1 ATP. Easy. Now in the second step, that's called as isomerization. If you know the basic chemistry, you will be able to tell what is isomers. What is isomers? They will have same, let me tell you, they will have something called as same chemical formula. Can you see? C6H11O9P. Same chemical formula, but the structure is going to become different. But different structural formula. Right? That is called as isomerization. So in the second step of glycolysis, your glucose 6-phosphate is getting isomerized to fructose 6-phosphate. Fructose 6-phosphate. Same. Only the structure is varying, the chemical nature is going to remain the same. The chemical formula is going to remain the same. The structure is varying right now. Isomerization. Now remember this point throughout your life. Whenever there is isomerization, there is no energy involvement. No energy is involved. No energy is involved here. No energy is involved. Remember this point in isomerization. Every single time there is isomerization, there is no energy is involved at all. Not all. Now, in the next step, fructose 6-phosphate. See, glucose to fructose, isomerization, no energy. Fructose 6-phosphate. This is the step right now. You see, fructose 6-phosphate. You have a phosphate at the 6th position. Now, to this particular structure, again you're adding a ATP. Again you're adding a ATP. Okay, we are adding a ATP. ATP is here. Now, what are we doing again? Same. This step is also phosphorylation. This step is also phosphorylation. We are taking a ATP, we are breaking the bond. This phosphate this time is attaching to the first position. First position. Now you tell me, this is called as fructose 6-phosphate. If we add one more phosphate here, what will it be called? Fructose 1,6. Simple basic chemistry, right? 1,6 bisphosphate. Fructose 1,6 bisphosphate. Does it require energy to formation of this? Yes, energy requirement is there. Yes, energy requirement is there. Do you see again? Energy requirement is there. See here. So this step is done. This step is done. And this step is done. Fructose 6-phosphate to fructose 1,6 bisphosphate. Keep this in mind. We had fructose 6-phosphate. To the first position, we are adding one more ATP. Phosphorylation. So fructose 1,6 bisphosphate is happening. First step and third step, ATP is being consumed. ATP is being consumed. Second step, isomerization. No need to buy out all of this. Visualize everything in your head. Okay. Now, we are at this step now. We are at this stage right now. We are at this stage right now. Now, what is happening here? What is happening here? Fructose 6-phosphate. Where is it? Fructose 1,6 bisphosphate is here. Okay, okay. We are here done till here. Till here we are done, right? Till here we are done. Fructose 1,6 bisphosphate. First step, second step, and third step. First step and third step, ATP is being consumed. ATP is being consumed. Second step, isomerization. Now, in the third step, what happens? In the third step, we have your. In the third step, what is happening? We had a 6-carbon molecule till now, right? 1, 2, 3, 4, 5, 6-carbon molecule. This 6-carbon molecule will split into 2. 1, 2, 3, 4, 5, 6. This 6-carbon molecule. This 6-carbon molecule will split into 2 3 carbons. 6 carbon is dividing into a 3-carbon molecule here and a 3-carbon molecule here. That's all. See here. 6 carbon is dividing into a 3-carbon molecule here and a 3-carbon molecule here. 3 carbon and 3 carbon. Simple. 3-carbon molecule, 3-carbon molecule. The first one is called as glyceraldehyde 3-phosphate. You can write this as a GAP. Or some of you write, NCERT writes PGAL. That is phosphoglyceraldehyde. Also can be written as PGAL. That is phosphoglyceraldehyde. The other one is DHAP. DHAP. Dihydroxyacetone phosphate. Dihydroxyacetone phosphate. DHAP. This is right now here is 3-carbon molecule. This right now here is a 3-carbon molecule. Here we had a 6-carbon molecule. Clear, all of you? Clear? So a 6-carbon molecule is split into a 3-carbon and 3-carbon. And the surprising thing is, your gap and DHAP, they are isomers again. They are what? Isomers. They are isomers. Now can they interchange? Yes, they can interchange, because their chemical nature is same, only the structure is different. Yes, they are isomers. So we do isomerization and we convert them. We do isomerization and we keep it, and we do isomerization, that is your DHAP is converted to gap, or you can also call it PGAL, that is glyceraldehyde phosphate, right? Or phosphoglyceric acid, phosphoglyceraldehyde or glyceraldehyde 3-phosphate, both same. Okay, so DHAP is getting converted to PG gap or PGAL. So by this step right now, till here we are done. Now. Any doubt, ask me. Till here, what happens till here? This step, what is happening in this step again? Let me repeat, if you should not have any confusion. DHAP is isomerized to gap. And that is the main reason, students. That is the main reason we have into 2 everywhere. Can you see into 2? Can you see? Everywhere from this step onwards, we have into 2, as well as everywhere into 2. Why do we think we have into 2? Because it's getting isomerized and everything we are considering into twice the number here. That's all. Twice the number. Okay, twice the number. DHAP is isomerized to gap. Now, what happens to the next step? What happens to your glyceraldehyde 3-phosphate now? Gap. What happens to the gap? From now, preparation is over. Till here, entire preparation is over. Till here, this one, entire prep time is over. This is prep time. This entire till here is your payoff stage. It's going to be your payoff stage. Now, what exactly is happening in your payoff stage? Now? Don't by heart this, students. Don't by heart this. We have your DHAP is getting isomerized to gap or PGAL. Now, your gap is undergoing oxidation. It's undergoing oxidation. Now, tell me quickly, remind me, what is oxidation? What is oxidation? Oxidation is nothing but loss of electrons or hydrogen. Yes. So your gap or PGAL is undergoing oxidation. That is the next step. See here, it's undergoing oxidation, PGAL. Now when it undergoes oxidation, as you know, it will release 2 electrons, 2 protons and energy. It is releasing redox equivalents. They are called as what? Redox equivalents. If you open your NCERT, the same line is mentioned. They are called as redox equivalents, electrons, protons and energy. Now what is the fate of them? These 2 protons, these 2 electrons and 2 protons are taken up by a new agent here. The new agent here is something called as NAD+. It's called as NAD+. That is nicotinamide adenine dinucleotide. Your NAD+ is taking up what? This NAD+ is taking all the electrons and protons given by the oxidation of GAP or PGAL. Now tell me, what is reduction? Reduction is nothing but gain of electrons or hydrogen? Yes. Now can I tell this NAD plus is undergoing reduction? Yes. NAD plus is undergoing reduction here, obviously undergoing reduction. Let's see what happens now. Well, let's see what happens. This NAD plus is undergoing reduction. It becomes NADH and H plus. NADH and H plus. NAD. So we had NAD plus, 2 electrons and protons, and finally we have the NADH and H plus. Now, what happens to this energy? Remember, we had electrons, protons, and energy. This energy is used to take an inorganic phosphate from the matrix, that is your cytoplasm, from the matrix, and it is attached to the first position here. This energy is utilized to take one inorganic phosphate from the matrix and attach to the first position here. So we had glyceraldehyde 3-phosphate. What will it become? Obviously it will become 1,3 now. 1,3 bis-phosphoglyceric acid now. Bis-phosphoglyceric acid. Are you understanding how it is happening now? Structure is very important, students. If you by heart this, very easy to by heart. But understanding requires a little bit of time. So what do we learn right now? We learned right now that your gap is converted to bis 1,3 bis-phosphoglyceric acid. Bis-phosphoglyceric acid right here, this step here. And in this stage, one NADH plus is produced. And how is it produced? This NADH and H plus is the result of reduction. It took the electrons and protons from GAP. GAP went under oxidation. Beautiful. This is a very beautiful concept, students. Very beautiful concept. It underwent oxidation. It released electrons and protons. That was taken up by your NAD plus, and we ended up getting NADH plus H plus. That's how we get this. That's how we obtain this right here. Beautiful, right? Beautiful. Clear? Let me summarize still here one more time. If you do not understand, let me summarize it for you. We have right now your glyceraldehyde 3-phosphate as a result of isomerization. This undergoes oxidation. Oxidation is what? Oxidation is a loss of electrons and loss of protons. It is your GAP is losing oxidation, losing proton and electron. Electron and proton and energy is released. This electron and proton are taken up by what? Taken up by your NAD plus. NAD plus undergoes reduction, because what is reduction? Reduction is nothing but gain of electron and hydrogen. Gain of electron or hydrogen. It's undergoing reduction to produce NADH and H plus. NADH plus H plus. Done. Now what about the energy? This energy is used to take one inorganic phosphate from the matrix and attach to the first position of your GAP. When we add one phosphate here to the GAP, it becomes 1,3. See, 1,3 bis-phosphoglyceric acid. Bis-phosphoglyceric acid. And we are at this step right now. Here. Done. Here we can see as triose phosphate, but it is the other name. Triose phosphate is nothing but your DHAP only. That is the other name for it. Triose is basically a common name for a 3-carbon compound with a phosphate. Triose phosphate. Triose, 3-carbon. Simple biology. Now we are at this step right now. Triose isomerase is the enzyme. But this is just triose phosphate here. Okay. Now we are at this stage right here. We have, what do we have right now? We have your 1,3 bisphosphoglyceric acid. Now 1,3 bisphosphoglyceric acid, we are adding something now. To this 1,3 bisglyceric acid, where ADP is added. ADP is added. Now students, in the starting of the chapter we learned, that where is the energy stored, students? Tell me, where is the energy stored? Energy stored in the form of bonds. Now this bond right here is unstable. Will this bond break? Yes. If this bond is broken, will this phosphate be released? Yes. If this bond is broken right now and this phosphate is released, this phosphate will attach to this ADP right here, this ADP. So one phosphate is lost here and this ADP will get converted to ATP. This ATP will get converted to ATP. Clear? So that's why we have what? And 1,3 bisphosphoglyceric acid, where this bond is broken, it gets converted to, it is 1,3, right? It was 1,3, it becomes 3-phosphoglyceric acid, just PGA. So it was 1,3 bisphosphoglyceric acid, it becomes 3-phosphoglyceric acid. Why only 3-phosphoglyceric acid? Why only 3? Because there is only one phosphate left at the third position. First phosphate is given to the formation of ATP. That's how we get the ATP. This step done right now. This step is done. So one ATP formation is happening here. ATP formation is happening. What do we have now? We have your 3-phosphoglyceric acid. 3-phosphoglyceric acid. Okay. 3-phosphoglyceric acid. 3 carbon. PGA. 3-phosphoglyceric acid. PGA. Okay. 3-phosphoglyceric acid. Now, again, what happens now? 3-phosphoglyceric acid to 2-phosphoglyceric acid. What is the difference, sir? Difference is very simple, students. This phosphate at the third position is sent to the second position. When the chemical structure remains the same, structure is varying. That is called as isomerization. So again, in the next step, isomerization is happening. Phosphate at the third position is sent to the second position. So we get 2-phosphoglyceric acid. And whenever there is isomerization, is there any release of energy? No. So no release of energy here. No release of energy, and we are at this step right now. Here. That is 2-phosphoglyceric acid. 2-phosphoglyceric acid. Beautiful. We are decoding the glycolysis, students. We are decoding the glycolysis. We are not mugging it up. Okay. Now what happens? There is a loss of water here. Water is lost, and we end up getting PEP. What is this PEP? The same PEP which we learned in your C4 cycle, phosphoenol pyruvate. It is phosphoenol pyruvate. What is lost? We end up getting phosphoenol pyruvate. Now again, students, again, this bond right here is unstable now. After water is gone, right? Water is gone. This is supposed to be phosphoenol pyruvate. This is not pyruvate. It is PEP. Phosphoenol pyruvate, this bond is broken. Now if this bond is broken, what happens to this phosphate? This phosphate is attached here again. This bond attached here. Now what do we get? So we end up getting just the pyruvate, and one ATP is produced. See here. Phosphoenol pyruvate, the bond is broken, energy is released, ATP's formation is happening, and we end up getting just pyruvic acid or pyruvate at the end. Pyruvate in the end. That is the end of your glycolysis. Where else? This is the time where you watch this lecture again if you want to truly understand the concepts. So if you have understood right now, yes, it will be in your memory. The only way you can solidify the memory is when you actually revise this. So after today's class, I want every student to come back to this video and check the video again and revise it one more time. Revise the entire step in one more time. Okay. First time you might be understanding right now, okay sir, I'm understanding everything, every step I'm following with you. But the only way you'll be able to solidify your memory is by revising it again and again. Okay, revise this concept again. And how I told you every single step what happens, I've told you. Every single step I've told you what happens. No by hearting. Okay, so we end up getting pyruvate. Now the next question is, what is the net profit? What is the net profit now? What is the net profit? I'll write it here. What is the net profit? What is the net profit? Students, in your preparatory stage, how many ATP was consumed? In your preparatory stage, can I write minus 2 ATP? Yes, minus 2 ATP. Minus 2 ATP. Preparatory stage. In your payoff stage, let me tell you. Here we have NADH. NADH is written. But do we only have one NADH or two NADH? Sir, obviously it will be into 2. Why into 2? Because everything from this point onwards is going to be into 2. Here they have shown just the list for 1. There will be a list here also, right? There will be a list here also, because this particular DHAP can also undergo glycolysis. If it is getting isomerization, everything will be 2 times here, 2 times. So obviously, if one NADH is produced from your triose phosphate or one glyceraldehyde 3-phosphate, only one. From DHAP also one will be produced. So everything is into 2. So 2 into 2. True. Right. So in payoff stage right here, we have 2 NADH. And one NADH produces 3 ATP. One NADH produces 3 ATP. So we have 2 NADH here. That is 3 into 2. That's going to be your 6 ATPs. One NADH produces how many ATP? One NADH produces 3 ATP. So it's going to be totally 6 ATP. Now what about in payoff stage again? How many ATPs produced? Here we have 1 ATP. Here we have 1 ATP. But is it only 1? No. Again it is into 2. Into 2. So 2 ATPs here and 2 ATPs here. So totally how many ATPs? In total 4 more ATPs here. 4 more ATPs in your payoff stage. Payoff stage 4 more ATP. Okay. 4 more ATP. Now what is the net ATP? 6 plus 4 is 10 ATP. But in preparation we have used 2 ATP. So minus 2 ATP. Minus 2 ATP. So we end up getting after subtracting we'll get 8 ATP from your glycolysis. We'll end up getting 8 ATP from your glycolysis. That is the end of your glycolysis. 8 ATP from your glycolysis. Beautiful, right? Beautiful chapter, right? Yes. 8 ATP. Now, what kind of question they can ask you? Apart from this, whatever they have taught you, they can ask you many different type of questions. For example, how the question will be asked? Obviously, they will never ask you the entire glycolysis. NEET examination they can ask. Board exam they can ask you. NEET examination they cannot ask you the entire glycolysis. They can ask you specific questions. Let me note down few for them. Let me tell you few of them. For example, they can ask you which step ATP is produced. Which step ATP is synthesized. ATP is synthesized, right? ATP synthesized. All of you know the first ATP synthesis is at 1,3 bis-phosphoglyceric acid conversion to 3-PGA. 3-PGA. Where is that now? Where is that now? That's the 7th step. 7th step. Where is that now? See here. 1,3 bis-phosphoglyceric acid to 3-phosphoglyceric acid. ATP is produced. That is the 7th step. Okay. The other one is where? Other one is your phosphoenol pyruvate to pyruvic acid. The other one is your phosphoenol pyruvate to pyruvate. That is going to be your last step. That is going to be your 10th step. That's going to be your 10th step. Easy. They can ask you, in which step NADH is produced? NADH or H plus is produced. They can ask you. So you should be able to tell it's produced at PGAL. PGAL. Remember, isomerization is going under isomerization, that is undergoing oxidation. Yes, PGAL to 1,3 BPG. 1,3 BPG. That is at the 6th step, right? See here. This is a PGAL to 1,3 bis phosphoglyceric acid. That is at the 6th step. 6th step. 6th step. Now, out of this particular reactions, which is irreversible step? Which is irreversible? Irreversible steps. Students, remember the irreversible steps are going to be your 1, 3 and 10. 1, 3 and 10. Irreversible step. Or they can ask you the rate determining step. Which is the rate determining step. The rate determining step is going to be your 3rd step. It's going to be your 3rd step. Clear? Or they can ask you, in which step phosphorylation is happening? Tell me, which step phosphorylation is happening? You should be able to tell me, sir, phosphorylation is happening at your 1st step as well as your 3rd step. As well as your 3rd step. Right? 1st step and 3rd step, phosphorylation is happening. Or they can ask you, in which step water is being removed? You should be able to tell me, sir, at the 8th step right here, 8th, 9th, 10th. At the 8th step here, water is being removed. Can you see? Water is being removed here. That is 2-phosphoglyceric acid to phosphoenol pyruvate. Water is being removed. Are you understanding? You should be able to tell all of these following points. Okay. Done. All of you. This is the way they can ask you questions in multiple way. Not just this, they can ask you multiple questions. Let me show you. See this question. See this question right here. Have a look at this question. It says that in plants, sucrose is converted to 2 molecules of monosaccharides due to the action of invertase, and then both enter the glycolytic pathway. Identify the monosaccharides. This is a NEET 2024 question. Didn't we just learn in your glycolysis, in the case of plants, sucrose is getting converted to glucose and fructose. Glucose and fructose. Easy question, right? Every question from this chapter is very easy if you understand the concept. Now, in glycolysis, the conversion of 1,3 your bis-phosphoglyceric acid to 3 PGA. Didn't we just learn 1,3 bis-phosphoglyceric acid to PGA, which is your technically the 7th step? 7th step, what happens? Energy yielding, energy utilizing, phosphorylation, isomerization. See, students, you should also know wherever isomerization is happening also. 1st step, 2nd step, isomerization, glucose to fructose. 2nd isomerization is where? 2nd isomerization is between your DHAP as well as your PGAL or GAP. The 3rd isomerization is between 3-phosphoglyceric acid to 2-phosphoglyceric acid. Do you have to by heart this? No. Understand the concept, you'll remember everything. Now tell me, 1,3 bis-phosphoglyceric acid to 3-phosphoglyceric acid. If you know here, ATP formation is happening, right? ATP formation is happening. Did we learn this? Bis-phosphoglyceric acid to 3-phosphoglyceric acid. ATP formation is happening. Yes. So what is the answer? Energy yielding process. ATP is produced. And this right here is again NEET 2024 question. Very easy question. If you understand the concept, everything is going to be easy. Look at this question now. ATP is used at 2 steps in glycolysis. Okay, 2 steps, all of you know. First step and third step, right? First step is used in converting glucose to glucose 6-phosphate, correct? And second ATP is used in converting fructose 6-phosphate into fructose 1,6-bisphosphate. In your mind, the entire structure should come. We have fructose 6-phosphate. To this fructose 6-phosphate, in the first position we are adding one more phosphate by using ATP. Okay, correct answer, correct answer. So assertion is true. Reason is explaining the assertion because at 2 places it's happening. So answer is obviously B. Correct explanation. It's correct explanation. Beautiful. Very easy. Look at this next question. Conversion of glucose to glucose 6-phosphate. The first irreversible step, action of glycolysis is catalyzed by. The first step, I told you, irreversible steps. There are 3 irreversible steps. 1, 3 and 10. This is the first one. Glucose to glucose 6-phosphate. The enzyme there was hexokinase. Didn't we remember hexokinase? We wrote it also there. So students, you need to remember the first and the last enzyme. Everything else is easy. The first and last enzyme. Let me write for you. The first enzyme here is going to be the first enzyme, hexokinase. Hexokinase. The last enzyme is going to be pyruvate kinase. Just remember these 2 enzymes. Remember these 2 enzymes. Pyruvate kinase, that's conversion of your phosphoenol pyruvate to your phosphoenol pyruvate to pyruvate with the release of one ATP. Okay. This is from your NEET 2019 question. Okay. Now till here, glycolysis is done. Glycolysis is done and dusted. I am deliberately going very slow so that every single student who is studying this chapter for the very first time does not by heart this chapter, in fact understand this chapter and try to, fall in love with the chapter. It's so technical, right? It is so technical and so nice. Oxidation, reduction, electrons are passing. Beautiful chapter. Beautiful chapter. Right, all of you? Now, what is the fate of your entire fate of your pyruvate? Now, what is the next step? For example, you're from your class 5th, you're coming to class 6th. You have so many new things, right? New fate, new encounters going to happen. Same way, your pyruvate has multiple fates. Pyruvate has multiple fates. For example, let me tell you. First we had the glucose. The glucose got converted to what? Glucose got converted to pyruvate. This step is called as what? This step is called as your glycolysis. Oh, I didn't mention one thing which is very important. Let me tell you that also. Where does it happen? It happens in the mitochondria. So it happens in the cytoplasm. Happens in the cytoplasm. Okay, happens in cytoplasm. Students, remember this point. It's going to be very important for you to remember. Your entire glycolysis is partial oxidation. It is partial oxidation. Now you might ask, sir, why is it called as partial oxidation? Because did you see anywhere release of CO2? Did you see? Did you see anywhere? No release of CO2 anywhere. No release of CO2 anywhere. That is why it's called as what? Partial oxidation. No release of CO2. Okay, partial oxidation. Beautiful. Now this particular pyruvate, how many carbon? 3 carbon. Glucose, 6 carbon. So technically, shouldn't we have everywhere N2? Yes, technically N2. Technically, you should be correct. Now, this pyruvate has 3 fates right now. 3 different fates. It can undergo 3 different fates. The first one second, students. Okay, the first two are in the absence of your alcohol. Absence of oxygen. Absence of oxygen. This is presence of oxygen. Okay, presence of oxygen. The first one is happening in your yeast. It's happening in your yeast. This is happening in your muscle cell. Muscle cell. The last one is happening in your mitochondria. Mitochondria. Now, what exactly happens? What exactly happens? In the case of absence of oxygen, it's called as anaerobic respiration. In the case of yeast, it will lead to alcoholic fermentation. Alcoholic fermentation. Beautiful. Now, what about this? In muscle cells, lactic acid fermentation. Lactic acid fermentation. This is your complete normal oxidation. Normal oxidation, that is CO2 plus water plus energy. Here we have lactate is produced, right? Lactate is produced. Lactate plus energy. Right, here we have ethanol. Ethanol plus energy. That is the entire fate. That is the entire fate of your pyruvate. Pyruvate under absence of oxygen, presence of oxygen. Absence of oxygen in the yeast, alcoholic fermentation. Absence of oxygen in muscle cell, lactic acid fermentation. In the case of mitochondria, CO2 plus H2O plus energy is produced. Energy is produced here. Okay, lactic acid fermentation. Very simple. Very simple. Now, let's focus on your anaerobic respiration now. That is lactic acid fermentation and alcoholic fermentation. For example, first we'll focus on your lactic acid fermentation. Students, important concept is going to come. Very important concept is coming. So please focus right now. What exactly happens in your lactic acid fermentation? Obviously. First of all, lactic acid fermentation happens in your muscle cells. It happens in your muscle cells. Or lactic acid bacteria. Lactic acid bacteria. Okay. And does it happen in presence of oxygen? No. It happens in absence of oxygen. Absence of oxygen. Okay. Absence of oxygen. Now, what exactly is happening to the pyruvate right now? What exactly? Your NADH is undergoing oxidation. What is oxidation, students? Tell me. Oxidation is obviously loss of electrons. Loss of electrons, right? So, electrons is lost. Electrons or protons are lost completely. These electrons or protons are taken up by your pyruvate. It is taken up by pyruvate. Now pyruvate is getting reduced. Exactly. This pyruvate is getting reduced. I mean, that is the line of your NCERT also. It is getting reduced to lactate. It's getting reduced to lactate. So it is getting oxidized to your NAD plus. See, NAD plus. Clear all of you now? Tell me on the chat quickly. This particular your NADH is undergoing oxidation. NAD plus undergoing oxidation. That is, it is losing the protons and losing the electrons, and it's getting converted to NAD plus. Now this electrons and protons are taken up by your pyruvate. Taken up by your pyruvate. Now pyruvate is getting reduced to lactate. Getting lactate. That's all. So only difference was I didn't mention the arrow mark here. So it got a little confusing. Okay. Clear all of you? And the enzyme here is going to be your, that is what they'll usually ask as a question. Lactate dehydrogenase is the enzyme. Lactate dehydrogenase is the enzyme. Clear? So your NADH is undergoing oxidation to release the loss of electrons, loss of protons are released. Those, that's why it's getting converted to NAD plus. Do you see? getting converted to your NAD plus. Now these electrons or protons are taken up by your pyruvate. So what is pyruvate now? Pyruvate is gaining proton and gaining electron. So it is getting reduced to lactate. It is getting reduced to lactate. Clear? That is the same line of NCERT, right? Let me show you the line of NCERT for that. See here. Can we go down? Friends, are you able to see the NCERT right now? See here. We have, when oxygen is inadequate, cellular respiration, pyruvic acid is reduced. See, pyruvic acid is reduced to lactic acid by lactate dehydrogenase. The reducing agent is NADH+, which is reoxidized to NAD+. Reoxidized, we wrote it properly, which is reoxidized to NAD+. This is the line of NCERT which we just discussed. Okay, getting reduced. Did all of you understand? So that is what we did here. Now, what happens here? Again, what happens in this case? In alcoholic fermentation. In the case of your alcoholic fermentation, again, we have your first step is decarboxylation. Decarboxylation. What is the decarboxylation? In from pyruvate, we are removing one carbon. One carbon is removed outside. Decarboxylation. So your 3 carbon is becoming 2 carbon, that is acetaldehyde. It is converted to acetaldehyde. Now again what is happening? Your NAD plus right here is losing electrons. It is undergoing oxidation. It is undergoing oxidation. What will it do? It will lose electrons and it will lose protons. Yes. Now these two electrons and protons will be taken up by your aldehyde. Yes, it will be taken up by aldehyde. Now this aldehyde will again be reduced to ethanol. Be again reduced to ethanol. Now what is reduction here? Reduction is what? Reduction is gain of protons and gain of electrons. It is gaining protons and electrons from here, from right here. Clear? From here. 2 electrons and 2 protons are taken up by your acetaldehyde and it is getting reduced to ethanol. It is getting reduced to ethanol. Clear? The enzyme here is again alcoholic dehydrogenase. So in your alcoholic fermentation, we have 2 enzymes, alcoholic dehydrogenase as well as your pyruvate decarboxylase. Clear? See, I don't want you to by heart this, understand the mechanism how it is happening. You first of all, decarboxylation is happening. 1 carbon is lost. We get acetaldehyde. Now this acetaldehyde is gaining protons and gaining electrons from where? Because of oxidation of NAD plus, NADH is getting oxidized to NAD plus is getting oxidized here, and acetaldehyde is reduced to ethanol. It is reduced to ethanol because it is getting protons and electrons from NAD plus. Clear? All of you? Yes, this is for class 12 chapter. Respiration in plants. Clear all of you? Understand this, your life will be easy. Okay. If you have any doubt, ask me right now quickly in the chat. Any doubt, quickly ask me in the chat. This is your entire alcoholic as well as lactic acid fermentation. Any doubt, quickly ask me. Clear, right? The reason I wrote oxidation here before was because that is happens in decarboxylation, oxidative decarboxylation, not here. Okay, it is undergoing reduction. Very simple it is right now. Now see here again what is happening. This is your entire anaerobic respiration. We have getting NADH is getting converted to NADH. Your entire NAD plus getting converted to NADH. That is, you know, glycolysis we learnt. Glycolysis. So we have 2 pathways. One is lactic acid, other one is alcoholic. Both cases, this NADH is getting utilized. The same NADH is getting utilized here. Getting utilized. Now tell me what is the net gain here? what could be the net gain of ATP then? Net ATP gain. Now obviously, if this particular, here glycolysis, in glycolysis first we have -2 ATP in your preparatory stage, then we have 2 NADH, then we have 4 ATP, right? This was in your glycolysis, Now if the same NADH is being utilized in the next steps, can we count this to NADH now in the total of your fermentation? No. Since it is being used completely, we cannot consider this. Now what are we left with? We are left with 4 minus 2, that's going to be 2 ATP. Just 2 ATP. Clear? Just 2 ATP is left. So this is going to be your entire net gain. 2 ATP is going to be the net gain of your lactic acid as well as alcoholic fermentation. Beautiful. Now, I want you to notice this one line here. Since this is only 2 ATP gained, is it more energy or less energy? Very less energy. In fact, it is just 7% energy. See here. In both lactic acid and alcoholic fermentation, not much energy is released. Less than 7% of energy in glucose is released, and not all of it is trapped as high energy bond of ATP. Okay, that is very important to understand. The other thing which you need to understand here is yeast poisoning. The yeast poison themselves to death when the concentration of alcohol reaches up to 13%. So if the yeast is making alcohol, if yeast is undergoing alcoholic fermentation, maximum alcohol content can be 13%. 13%, that's all. If you want higher concentration of alcohol in your drink, distillation has to undergo. Okay, distillation has to undergo. Look at this question now. Look at this question right here. From your, it's a NEET PYQ again. It's a NEET PYQ. NEET PYQ again. The amount of energy is released from glucose during lactic acid fermentation. Obviously, we just learned it is less than 7%. Right? Now, that was the end of anaerobic respiration. Clear? Now, can we start with your aerobic respiration? Did all of you understand glycolysis? Don't by heart. Did all of you understand alcoholic fermentation and lactic acid fermentation? Very easy to understand. It just requires a little bit of practice. If you by heart it, you will forget it. I'm telling you, if you by heart it, you will forget it. Okay? Make a logic out of it. Now, in aerobic respiration, what happens in aerobic respiration? This is your complete aerobic respiration. In aerobic respiration, whatever we had the pyruvate, or whichever we had the pyruvate in the cytoplasm, the pyruvate in the cytoplasm is sent to the mitochondria now. The next step, aerobic respiration, happens in the mitochondria. So pyruvate is now where? Pyruvate is inside the mitochondria now. Pyruvate is finally inside the mitochondria. Now what happens in your mitochondria? The first one is called as your oxidative decarboxylation, also called as a link reaction. Then we have Krebs cycle. Then we have something called as oxidative phosphorylation. 3 different steps are happening inside the mitochondria. The first one is called as your oxidative decarboxylation, mitochondrial matrix, Krebs cycle, mitochondrial matrix, electron transport chain, that is electron transport system, crestae. It happens in your inner mitochondrial matrix. Crestae or inner mitochondrial matrix. Now, let's understand what is the meaning of oxidative decarboxylation, also called as your link reaction, also called as your gateway reaction. Now this reaction is a reaction which is present between your glycolysis as well as your Krebs cycle. Between glycolysis and Krebs cycle, between reaction is called as link reaction. Okay. Now what happens here? Obviously we have the pyruvate first. We have the pyruvate which is entering into the mitochondria. The first step is called as decarboxylation. That is minus CO2. minus CO2. So can you see? From pyruvate molecule, 3 carbon molecule, if you remove one CO2, what is remaining? What is remaining? If you remove the CO2 molecule, you're only left with acetate or acetyl group. Acetyl group. You're only left with acetyl group. Basic chemistry. Basic chemistry. That is COCH3. COCH3. Right? COCH3. COCH3 is left. Now this acetyl group undergoes oxidation. This acetyl group undergoes oxidation. Now what is oxidation? Tell me quickly in the chat. What is oxidation? Oxidation is loss of electrons and loss of proton. Now this acetyl group is losing protons and losing electrons. Okay. It is losing protons and losing electrons. Electrons are being lost and protons are being lost. Now this electrons and protons are taken up by, they're taken up by your NAD+. Electrons and protons are taken up by NAD+. So NAD+ is taking protons and electrons and getting converted to NADH and H+. So technically, what is happening here? Reduction is happening. Reduction is happening. Reduction is happening. Okay, so your acetyl group is undergoing oxidation. Electrons and protons are lost. Those electrons and protons are taken up by your NAD+. It is getting reduced to NADH and H+. NADH and H+. Now to this acetyl group, we are also adding a coenzyme group. A coenzyme is also added to the acetyl group. And finally we get what? Acetyl-CoA. What do we get? Acetyl-CoA. Clear? We get acetyl-CoA. Beautiful, right? Beautiful. This is called as oxidative. Why? Because oxidation is happening. Decarboxylation, because CO2 is being lost in the first step. So CO2 is lost, we get acetyl group. Acetyl group undergoes oxidation. When it undergoes oxidation, H+ and electrons are lost. Those H+ and electrons are taken up by your NAD+. This NAD+ is getting reduced to NADH and H+. That is why we tell you, in your link reaction there is release of what? In your link reaction there is release of NADH. There is release of NADH. And we find, and there is also addition of coenzyme, and we end up getting acetyl-CoA. 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 Beautiful. The enzyme here is very important again. Let me show you. The enzyme here is pyruvate dehydrogenase enzyme, also Mg2+. Also Mg2+. Pyruvic acid plus coenzyme plus NAD+, we end up getting acetyl-CoA, CO2 as a byproduct. Remember, decarboxylation, right? Decarboxylation, and NADH and H+ we are obtaining. NADH and H+ we are obtaining. You should understand how its CO2 is coming out, how this NADH is being produced actually, how it is being produced. Okay, that is your entire step of your link reaction. Now this acetyl-CoA enters your Krebs cycle now. It finally enters the Krebs cycle. Enters the Krebs cycle. Who gave the Krebs cycle? Krebs cycle was given by Hans Krebs. It was given by Hans Krebs. Hans Krebs. Okay, Hans Krebs. Now, can we study the entire Krebs cycle now? Can we start with the Krebs cycle now? Can we start? Can we start the Krebs cycle now, students? Tell me how many NADH is produced here? How many NADH is produced here? For one glucose, obviously, since here we've written only one pyruvic acid, that's why we've written only one NADH here. But in reality, we have how many pyruvate? 2 pyruvate from one glucose. So we'll end up having 2 NADH. We'll end up having 2 NADH. Not 1, 2 NADH. Okay, 2 NADH. Now, the next thing is going to be your Krebs cycle. How many of you find Krebs cycle difficult? Tell me in the chat quickly. How many of you find Krebs cycle difficult to understand and not just by heart it up? Tell me, honestly tell me in the chat. How many of you think Krebs cycle is very difficult to understand? Sir, I'm never understood Krebs cycle. Tell me, honestly tell me in the chat. So, can I give you a small trick to remember? It's the same trick which I always use. Same trick which I always use. So, I'm going to use the same trick only, obviously. Why will I change my tricks? The trick is very simple, students. The trick is very simple. The trick is very simple. That is, officer, can I keep selling sandwiches for money? That's all. That's all. Officer, can I keep selling sandwiches for money? That is the trick. Okay, that is the small trick. Now, all of you are done with me. All of you are done with me. First, we have the oxaloacetate. First, we have the oxaloacetate. Oxaloacetate. That is O. O for oxaloacetate. This oxaloacetate will get converted to C for citrate. C for citrate. Now, this oxaloacetate is actually a 4-carbon compound. This oxaloacetate is a 4-carbon compound. What is entering the Krebs cycle? What did we learn? Into your Krebs cycle, acetyl-CoA is entering, right? From a link reaction, acetyl-CoA is entering the Krebs cycle. Acetyl-CoA is a 2-carbon compound. 2-carbon compound. 4 plus 2 is going to be 6-carbon compound. 6-carbon compound. Citrate. Citrate 2. Isocitrate. Citrate 2. Isocitrate. Isocitrate 2. Alpha keto glutarate. Alpha keto glutarate K. Then S. Then we have S, that is succinyl coenzyme A. Succinyl coenzyme A. Then yes, that is succinate. That's going to be succinate. Succinate. From succinate, what do we have? F. F is fumarate. Fumarate. After fumarate, what do we have? M. M for malate. From malate, again we go back to oxaloacetate. Again we go back to oxaloacetate. Now we need to fill a few things here. Fill a few things here. Okay. Now here, isocitrate to alpha ketoglutarate, you need to know the first oxidative decarboxylation happens here. The first oxidative decarboxylation happens here. So in this, if it is oxidative decarboxylation, should there be release in carbon number? Decarboxylation? Yes, sir. So this is again 6 carbon. This is 5 carbon. It becomes 5 carbon. In oxidative decarboxylation, what happens, students? We learned here, right? We learned here. This is oxidative decarboxylation, right? This was oxidative decarboxylation. In oxidative decarboxylation, there is release of CO2 and also formation of NADH. NADH. So there is release of CO2 and also release of CO2 as well as formation of NADH plus H plus. Again. Now this is this place where we have second oxidative decarboxylation. Again. So this is 5 carbon. So this should become 4 carbon. Why 4 carbon? Because again, one CO2 is released out. Again, in oxidative decarboxylation, there is NADH formation plus H plus. That's all. That's all. Now, succinyl coenzyme to succinate, there is formation of GTP. In case of your animals, ATP in case of your plants. GTP. This GTP becomes ATP later on. Doesn't matter. Now, succinate to fumarate. Succinate to fumarate, there is formation of your FADH2. There is formation of FADH2. Now, fumarate to malate, there is entry of water. Now, malate to oxaloacetate, again, there is formation of NADH2. But is this decarboxylation? Is this last part decarboxylation, oxydecarboxylation? No. Why it is not oxydecarboxylation? Because there is no release of CO2. See, in oxydecarboxylation, there is release of CO2. Oxidative decarboxylation. But here it is just oxidation. Okay, it is just oxidation here. Just oxidation. So this was 4 carbon, and 4 carbon. 2 carbon coming, 6 carbon. That is your entire Krebs cycle, friends. That's it. Done. Krebs cycle is done. That's all. Look at it. All the questions which you have in your examination comes from this particular slide here. That's all. Nothing is there. Now, do you want to understand in detail in every step now? That is my question. Every single step will go one by one right now, and we'll decode it. How we decoded your glycolysis, right? In similar fashion, let's try to decode this particular part also here. Completely decode it. For example, in first step what happens? Okay. Now, what happens in the first step? That is, acetyl-CoA is entering, I told you. Acetyl-CoA is entering, and acetyl-CoA is added with oxaloacetate. That is, we know that oxaloacetate here. Acetyl-CoA plus oxaloacetate will result in citrate. Citric acid. The reason this is the first product, right? This is the first stable product of your Krebs cycle. That is why your Krebs cycle is also called as what? Krebs cycle is also called as your citric acid cycle. Okay, so what is happening here? Here water is also entering. Your oxaloacetate is getting converted to citrate. Same carbon number, 4 plus 2, 6 carbon. This CoA enzyme from here, from your acetyl-CoA, the CoA part is removed. And it is sent back to your link reaction. Gone. And the enzyme here is citrate synthase. Synthesis of citrate. So we finally have the citrate. Now, what happens to citrate, student? This is your citrate formation, which is your 6 carbon molecule. Okay. Now, citrate to isocitrate. Now, this is again isomerization. In isomerization, is there any release of energy? No. Gain of energy? No. Isomerization. So, citrate to isocitrate. Enzyme aconitase, you don't have to remember this. Isocitrate. Now, isocitrate to alpha ketoglutarate is the what? It is the first oxidative decarboxylation. Let me explain again one more time what is oxidative decarboxylation. So listen to me very carefully. What happens here? You have your citrate and we have alpha ketoglutarate. Citrate and alpha ketoglutarate, all of you know. From this citrate, obviously, since it is oxidative decarboxylation, there is release of CO2. Yes, there is release of CO2. And that is why isocitrate, which is a 6 carbon here, 6 carbon is becoming 5 carbon. Done. Now, this particular product is undergoing oxidation. Why it's called as oxidative decarboxylation? If isocitrate is undergoing oxidation, what will happen? Loss of electrons or gain of electrons? Oxidation is loss of electrons. It is losing electrons. Electrons or protons. Protons are taken up. Now this electrons and protons are taken up by whom? This electrons and protons are taken up by NAD+. They're taken up by NAD+. And NAD+ will undergo reduction. Gain of protons and gain of electrons. It will get converted to NADH. Done. That is called as your oxidative decarboxylation. And it is happening only 2 times in your entire cycle. First time between isocitrate to alpha-ketoglutarate, and second time between your alpha-ketoglutarate to succinyl coenzyme A. Now here we have 4 carbon, this becomes again 3 carbon because loss of CO2. Loss of CO2. See here. Loss of CO2. Loss of CO2. So we have succinyl coenzyme A. From succinyl coenzyme to succinate, there is formation of a GTP. That is, this technically is not oxidation right now. Listen to me very carefully. This step right here in the glycolysis, again this is a question. This step right here, that is between your succinyl coenzyme A and succinate, is actually phosphorylation. It is the only phosphorylation. The only phosphorylation which is happening in your entire Krebs cycle. Rest everything is oxidation. Oxidation is happening. And protease form. NADPH formation, FADH formation. Right? All that is result of oxidation. But here it is the result of your phosphorylation. See, phosphate is added to here. We get a succinyl phosphate. And this phosphate is removed again. This phosphate is added to GTP. And we end up getting GTP. So this phosphate is added to the GDP, we get GTP, and we get the succinate. So this is a question, students. In how many times in your Krebs cycle, in one Krebs cycle, phosphorylation is happening? Only once. That is at this step right here, phosphorylation. And it is the only step. In which oxidation is not happening. All of you will clear that. That point right there, right, is a NEET 2024 PYQ. If you by heart this, you will not understand. That right here is a NEET PYQ 2024, this year PYQ, previous year question. So this step right here, succinyl coenzyme to succinate, is the phosphorylation, and it is not oxidation. Okay, not oxidation. It is happening only once. In one cycle, it is happening only once. In one cycle, there is only, for one pyruvate, in only one cycle, it is happening only once. One GTP formation. Okay. Now here we have succinate to fumarate, that is FADH2 formation, that is by oxidation. It is undergoing oxidation. Electrons and protons are produced. That is taken up by your FAD+. It is getting converted to FADH2. Same happens in the case of your here malate to oxaloacetate. It is just oxidation. It is not oxide-decarboxylation. Just oxidation. Okay. See here. FAD. This is undergoing oxidation. Electrons and protons are released. Electrons and protons are taken up by FAD, getting converted to FADH2. The same happens in the last step. The same happens in the last step also. The same thing happens in the last step. Oxidation is happening. This malate gets oxidized to oxaloacetate. When it undergoes oxidation, electrons and protons are released. They are taken up by your NADH2. They are taken up by NAD+. They are taken up by NAD+, getting converted to NADH. Clear, all of you? Clear? I know, students, no one on YouTube will ever teach you this detail. No one ever. If you go check any video out there, this detailed Krebs cycle, this concept wise, very few people can teach you this way. Okay? Because when the questions come, you will understand the importance of concept. Okay? So here if you look at the reaction of your Krebs cycle, it says pyruvic acid plus 4 NAD plus. 4 NAD plus. Let me break it down. Let me build on this. Here, two of them are for oxidative decarboxylation, two of them. One is for just oxidation in the Krebs cycle. The other one is for link reaction. Yes, they have compiled your link reaction. Yes, they have compiled your link reaction in this reaction only. We have one FADH, 2 water is being released, ADP and inorganic phosphate. Mitochondrial matrix, we get 3 CO2. Do you see 3 CO2? But did we just learn in your entire Krebs cycle there is only 2 CO2? Yes. So here we have again oxidative decarboxylation 1, This is from your Krebs cycle. This one is from link reaction. One is from your link reaction. Because remember, in your link reaction also, oxidative decarboxylation takes place. Same thing happens there also. Okay. So we end up getting 4 NADH, 3 from Krebs cycle, 1 from link reaction. FADH to 1 ATP or 1 GTP. 1 ATP or 1 GTP. Done. Look at this question from your NEET 2024. If you by heart, you will understand this. It says that identify the step in your tricarboxylic acid cycle, also called as your citric acid cycle or Krebs cycle, tricarboxy cycle. I didn't tell you why it's called tricarboxy, right? Why is it called as tricarboxy? Some of you might ask me, sir, why is it called as tricarboxy, sir? Tricarboxy. For that, you should see the structure of your citrate. Look at the structure of citrate. You will understand why it's called as tricarboxy. See here. How many carboxyl group is there? If you look at the structure of your citrate, 1 carboxyl group, 1 more here, and 1 more here. There are 3 carboxylic groups here. So that's why it's called as tricarboxylic cycle. Did you understand? It's called as tricarboxylic acid cycle. 3 carboxyl groups. 3 carboxyl groups. Okay. See here. In citric acid cycle, tricarboxy cycle or Krebs cycle, which does not involve oxidation of substrates. Oxidation of substrate is not happening where? Where is not happening oxidation? I told you in only one step oxidation is not happening. Only in one step oxidation is not happening, I told you, right? See here, oxidation. Citrate to isocitrate, alpha ketoglutarate, oxidation happening. Alpha ketoglutarate to succinyl, oxidation is happening. But between your succinyl coenzyme A to succinate, between succinyl coenzyme to succinate, what is happening? Phosphorylation is happening. What is happening? Phosphorylation is happening. Phosphorylation. Succinyl coenzyme to succinate. Phosphorylation is happening. Oxidation is not happening. Concept question, right? Concept question. Did you understand how important it is to understand the depth of every concept, not just by heart it from the outer covering, learn one mnemonic, learn in the mnemonic, by heart Krebs cycle and just go. This is a proper concept-based question from your NEET 2024. NEET 2024. It says NCERT, from NCERT itself. Understand? Okay. See, remember, malic acid to oxalic acid, what is happening? Malic acid to oxalic acid, there is formation of NADH, right? Formation of NADH. But is this oxidative decarboxylation? No. It is not oxidative decarboxylation. It is just oxidation. If you don't remember this step right here. Here. Malic to oxalic acid, NADH is being produced. Normal oxidation. Normal oxidation. Look at this question now. Look at this question. How many times decarboxylation occurs during each TCA cycle? Last year question. How many times it's happening in each TCA cycle? Obviously, you know it's happening twice. Because 2 times oxidative decarboxylation happening. 2 times oxidative decarboxylation is happening. That's what's written. Answer is 2. Answer is 2. The 5-carbon compound formed during the TCA. We just now learnt 6-carbon citrate. First oxidative decarboxylation results in a 5-carbon compound. That 5-carbon compound, which is result of first oxidative decarboxylation, that is nothing but your alpha ketoglutarate. 5-carbon compound. The number of substrate level of phosphorylation in one turn of citric acid cycle is citric acid cycle. Number of phosphorylation. That's going to be 1. Only 1 GTP is being produced. Only 1 GTP is produced. Phosphorylation, we learnt phosphorylation. Only once it is happening. 1 GTP is produced in one citric acid cycle. Clear? I believe there should be no doubt till here, right? So do you understand the importance of concepts, students, now? Importance of concepts. How every concept plays a key role in learning it, right? So it's been 2 hours since we start teaching. Can we take a 15 minutes break and come back now? Can we? My throat is hurting a little. From 2 hours we are learning this chapter. You're only left with 2 concepts. You're only left with 2 concepts. That is electron transport chain as well as your amphiboic pathway. That's all. Only 2 concepts left. The chapter is over. Nothing is there in this chapter. Students. Okay? Let's take a 15 minutes class and we will resume the class at 7:20. Let's 7:25. It's almost 7:10, right? 7:25 class resumes. Okay? 7:25. Okay, students? I'll just go drink some water and I will just sit for some time. My legs are hurting a little. 2 hours. Okay? 2 hours you can't even watch a movie, so you are learning something. Okay? So that's great for you. So I'll be back. Okay? Welcome back all of you. Let's go towards the end of the chapter now. The chapter is almost over and I believe we can finish it very easily. The only concept which is going to be a little difficult for you to understand is going to be your electron transport system. Again, if you draw and understand, it's going to be very easy. If you draw and understand, it's going to be very easy, and questions, we'll do the questions. Questions, we will do the questions. Don't worry. Now, didn't we just learn that, yes, in your Krebs cycle, there is a release of NADH. In your Krebs cycle, there is a release of NADH, there is a release of FADH, and also in your link reaction, there is a release of NADH. Now, what is the fate of this NADH and FADH? Where is the utilization and where are they used in your energy generation? And where is the oxygen actually used? We've been telling, right? We've been telling that, what about oxygen? Where is it used actually? Did you see anywhere usage of oxygen anywhere till now? No. So what is the actual use of oxygen, which we told in the starting of the class, where I told you oxygen is entering and oxidizing the food? Where is the actual use of oxygen? All those answers will be discussed in your electron transport system, which is also called as your oxidative phosphorylation. Oxidative phosphorylation. NADH and FADH2 are transferred to the electron transport chain. ATP is formed as a result of transferring electrons from NADH and FADH2. So from NADH and FADH2, we are obtaining the electrons and protons. Electrons as well as protons. Okay. So where does it happen? First of all, see, this is the matrix. This is the matrix. Inside the matrix, we have link reaction and Krebs cycle. Link reaction and Krebs cycle. But do you see this foldings and the inner membrane? See, students, all of you know your mitochondria has two layers. This is the outer membrane and this is the inner membrane. So on this inner membrane, that is inner mitochondrial membrane, we have the electron transport system. Inner mitochondrial membrane. Inner membrane. Do you see this? Inner membrane. That's where the electron transport system works. That's where the entire electron transport system works. So if you want to understand the electron transport system, you need to know something about your protein complexes as well as your mobile carriers. Protein complexes as well as your mobile carriers. In total of 5 protein complexes and we have 2 mobile carriers. What is a protein complex? Protein complex is a complex made up of proteins and electron carriers as well as your electron carriers or metal ions. They're made up of protein as well as metal ions. Metal ions. There are many metal ions also. Metal ions are there. Okay, electron carriers. We have complex number 1 called as NADH dehydrogenase. Complex number 2 called as succinate dehydrogenase. The same enzyme is present in your Krebs cycle also. Yes. For your succinate to succinate, for your FADH formation. Then we have cytochrome BC1 complex, cytochrome oxidase, ATP synthase. That is going to be your complex number 5. We have also something called as, everything is last, then we have your ubiquinone and cytochrome C. We have cytochrome C. We have 2 different complexes that is called as mobile carriers. Mobile carrier basically like taxi. They are like taxi. You take your electrons, add it to the taxi and they will take it and go. For example, we have your ubiquinone. We have the ubiquinone, right? One second, students. This is your ubiquinone. It's called as UQ. Now this UQ, whenever it takes electron, it becomes, or proton, it becomes UQH2. That is ubiquinol. That becomes ubiquinol. It becomes ubiquinol. Remember that. Ubiquinol. Okay. Then we have cytochrome C, which will tell you where it is requirement. But before that, can all of you draw this entire structure once? I'll give you, I should have shown this, but can you all of you draw this? Different complexes? Different complexes? Can you draw this? Yes. This is going to, see, if you draw it, then only you will understand this. If you don't draw it, not going to understand, honestly. This is going to be your complex number 1. Complex number 1. This is going to be your complex number 3. Here we have complex number 2, complex number 4, and finally complex number 5. Oops. Complex number 5. Complex number 5. Now what is this? This region is your matrix region. Matrix region. This is where Krebs cycle is taking place. Yes. Krebs cycle is taking place here in the matrix region. In the matrix region. Okay. Now what exactly happens in the first system? What happens technically in the first mitochondrial first system? In the first system, what happens is here. Is it visible, students? Is it visible? I hope it is visible. If not, I'll use red. Now before we let's fill up each complex. Let's fill up each complex right now. Let's fill it up. Let's fill the first one. We have your F, M, N as well as your iron sulfur centers. Iron sulfur centers. This is called as cytochrome B7 complex. Cytochrome B, cytochrome C. Cytochrome C as well as iron sulfur centers. And this is cytochrome FAD. We have F, iron sulfur centers, FAD, iron sulfur centers and FAD. This is cytochrome B7 complex, copper center A, copper center B, cytochrome A1, cytochrome A3. This is F0 and F1. This is going to be your F0 and F1. F0 and F1. Very simple, very simple. Now, what happens in your complex one? This is your intermembrane space. This is your intermembrane space. Intermembrane space, that is between the two membranes, between the two membranes, outer membrane, inner membrane. That is called as intermembrane space. This right here, intermembrane space. Okay. This is inner mitochondrial membrane, that is matrix. Inside inner mitochondrial membrane, you have intermembrane space. So in the first complex, we have your NADH2 getting converted to NAD+. Now tell me, is there any loss of electrons and loss of protons? Yes, electrons are being lost. Electrons are being lost here. Yes, sir. Electrons are being lost. 2 electrons as well as 2 electrons are sent. Okay. Now what about protons? What about the protons? Now these 2 protons here will be sent to the intermembrane space. 2H plus. For this particular complex, NADH dehydrogenase works on its own. It works on its own. C1 NADH dehydrogenase works on its own, and it takes 2 protons from your matrix region and also sends it to your intermembrane. So again 2H plus. The particular complex is also working on its own without any type of your NADH. It's working on its own. This cytochrome, your complex number 1 NADH dehydrogenase is taking 2H plus from the matrix and it is sending. So how many have in total? In total we have 4H plus here. 4H plus here. Done. Now what about your, let's come back to this complex later on. What about this complex right here? What about your complex number 2? Complex number 2 has another name also. The complex number 2 called as succinate dehydrogenase can also be called as your FADH2 dehydrogenase. Okay. Now here what is happening here? Here FADH2 is getting reduced, getting oxidized, loss of protons to FAD plus. It's getting oxidized, loss of electrons and loss of protons. Yes. So here also can I tell electrons are entering inside? Electrons are entering. 2 electrons. Okay. Now from here the electrons are taken up by. Something called as UQ. The mobile carrier. Beautiful. Here also UQ. Now this UQ after taking electron becomes UQH2. Becomes UQH2. H2. Now you might ask, sir, where is the H2 coming from? Where is the H2 coming from? Very good question. This ubiquinone takes 2H plus from the matrix. This ubiquinone takes 2H plus from the matrix and it gives it to here. It gives it here. And the cycle continues. And the cycle continues. Okay. 2H plus, it gives 2H plus to the UQ, and the UQ is, see, it becomes UQ becomes UQH2. Here also becomes UQH2. But is this ubiquinone taking 2 protons from here? No. These 2 protons, do you see? These 2 protons right here. Here. These 2 protons are taken up by your UQ. These 2 protons are taken up by UQ. Let me use a different color so you can understand it better. Here. These 2 H2 is taken by UQ. Taken by UQ. 2 protons. Same 2 protons are here. 2 protons here. Electrons are being circulated. Electrons are being circulated. 2 electrons. 2 electrons. 2 electrons. But 1 electron is given to your third complex. 1 electron is given to the 1 complex. Okay. It's given there. Now what about the 2 protons here? Yes. The 2H plus is also given here. Same 2H plus is also given here. Given. Okay. 2H plus is given to your third complex, 2H plus is given to third complex. Now, you might ask me, sir, here H2 was directly sent to your intermembrane space. Why can't we send this H2 directly to intermembrane space? You might ask me. Answer is very simple. Because your complex 1, complex 3 and complex 4 and complex 5 are your intermembrane proteins. That is, can you see? They're all the way to the intermembrane. Intermembrane. Can you see intermembrane? But your complex number 2. Your complex number 2 is never entering the intermembrane. Your complex number 2 is a inner membrane protein. Inner membrane protein. It's for part of your inner membrane. Complex number 2 cannot come out. Can you see? It's only stuck till here. So that's why this H2 is given to UQ. Beautiful. Now, one more question might arise in your mind. Sir, here 2 electrons are entering. 2 electrons are entering. Why only 1 electron is being sent from each side? Why 1 electron is being sent? The reason is very simple. The reason is very simple because you need to make sure the entire cycle continues. You need to make sure the cycle keeps on continuing. So 1 electron is consumed in the continuing the cycle, to continue the cycle. Okay, continue the cycle. So now you tell me how many H plus are there totally? 2H plus from here, 2H plus from here. So 4H plus are sent here. 4H plus are sent here. Easy. Okay, 4H plus are sent here. Now what happens? Now this 4H plus has to be transported to your complex 4. Complex 3 to complex 4 they has to go. It needs to take a taxi. The taxi is very simple. The taxi is here already. The taxi is here. The taxi is here. The taxi is called as cytochrome C is a taxi. Cytochrome T is a taxi. So these 2 electrons will be sent to your taxi. This taxi will reach here. This taxi will reach here and electrons are sent. Electrons are being sent up. Yes. Electrons are being sent up. 2 electrons. 2 electrons. 2 electrons. 2 electrons. Now, throughout the class, you've been asking me, sir, what is the use of your what is the use of oxygen? Where is it even useful? Right here. Right here. This final electron acceptor, the final electron acceptor in your entire electron transport chain is none other than oxygen. Is none other than your oxygen. So main work of oxygen is saved until the very end. Till the very end there is no work of oxygen. This is the actual use of oxygen, and water is formed as a byproduct. Water is formed. Beautiful. Now when this is happening from the matrix region, 2H plus is sent to the intermembrane. 2H plus is sent to intermembrane. 2H plus is sent to the intermembrane. So how many H plus do we have here in total now? 4 plus 4 plus 2. That's going to be your 10 H plus is here now. Totally 10 H plus is here. Now this 10 H plus will be sent up. 10 H plus sent up. That is called as proton gradient formation. Do we have a gradient now? Yes, we have a proton gradient, means proton difference is there. Proton difference. So this 10 H plus will be sent up. Will they be sent together 10 in a bunch? No. They will be sent in groups. They will be sent in groups. According to your new NCERT, 4H plus will be sent at once. When the 4H plus is sent at once, it makes 1 ATP. It makes 1 ATP. It makes 1 ATP. 4H plus are going up and they are making 1 ATP. 1 ATP is produced. 4H plus. 3H plus to rotate the ring. 1H plus to bring the phosphate catalytic activity. That is your entire electron transport chain. Done. Done. That's all. Electron transport chain is over. Done and dusted. That's all is the electron transport. What is there? That's all. This is your entire electron transport chain. This is according to your new NCERT. This is according to your new NCERT. 4H plus provides 1 ATP. Done. All of you can take a photo of this. Don't worry, the entire notes will be available to you on the Telegram channel. That is Basavaraj Sir Biology. Entire notes will be available. Entire notes will be available. This is your electron transport chain, students. And if you don't follow NCERT for this. Do not follow NCERT for this. Diagram you can follow, not the writing part. They are not matching. They are not matching. See here. We have 4, 4, 2. Complex 1 NADH. Complex 3, complex 2, complex 4 and complex 5. What is there? The same thing which we wrote. UQ to UQH2, UQ to UQH2. Electrons are being sent here, proton is being sent here. Same thing which we discussed. Cytochrome C, here, cytochrome C. Done. That is your entire chapter. Now, let's do a respiratory balance sheet. Let's do a respiratory balance sheet. What is the total ATP? Like, total ATP which we obtain from your entire glycolysis. Entire glycolysis, how many ATP did we obtain? First, we'll account for glycolysis. First, for glycolysis. Obviously, in the case of glycolysis, minus 2 ATP in your preparatory stage. Let me write that first. In preparatory stage, it was minus 2 ATP. Minus 2 ATP. In payoff stage, we had 2 NADH, which gave us around 6 ATP. 6 ATP. We also had 4 ATP as substrate level phosphorylation. In total, we obtained 6 plus 4 was 10. 10 minus 2 was 8 ATP. 8 ATP was obtained. Done. Now, second was link reaction. Link reaction, gateway reaction. In gateway reaction, there was oxidative decarboxylation and result was 2 NADH. 2 NADH. That is nothing but your 1 NADH provides 3 ATP. 3 into 2 is going to be 6 ATP. 6 ATP. Clear? The third one is going to be your Krebs cycle. Krebs cycle. In your Krebs cycle, there are 3 NADH in one, that is in one cycle there is 3 NADH. In one cycle there is 3 NADH, but remember it is for entire 1 glucose. So 1 glucose means 2 pyruvate. Then the cycle is happening 2 times. Cycle is happening 2 times. See, if it was only 1 pyruvate, 3 NADH. Cycle is happening 2 times. So this will be how many ATP? This will be 6 NADH. In one cycle there is 3, but for 1 glucose it is happening twice, right? 2 pyruvates. So 6 NADH. 6 NADH 2 GTP, 2 GTP as well as 2 FADH. And your 1 NADH provides 3 ATP. 3 2s are, say 6 3s are 18 ATP. Two ATP here. 1 FADH provides 2 ATP. 2 2s is 4. Clear? What do we have now? 18 plus 2 is 20. 20 plus 4 is 24 ATP here. 24 ATP here. We have 6 here. 6 ATP. And we have 6 here. So we have 8 here. We have 8 ATP here. 8 ATP. Now what is the final? 24 plus 6 is 30. 30 plus 8 is 38 ATP. 38 ATP is the final answer. 38 ATP is the final answer. Done. Done, 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 done. 38 ATP is the respiratory balance sheet. This is your entire respiratory balance sheet of your entire for one glucose. Look at this question now. Look at this question. The question says that match the following. Where does it happen? Citric acid cycle, all of you know it happens in your mitochondrial matrix. Glycolysis happens in your cytoplasm. Yes. Electron transport chain happens in your inner mitochondrial membrane. Proton gradient is found where in the intermembrane space of mitochondria. Here, right? Here. Proton gradient is found in your intermembrane. It is found in your intermembrane. So what is the answer? A is 2, P is 1 here. This is the answer. This is your NEET 2024 question. NEET 2024 question. PYQ. NEET 2024 PYQ this is. What is the answer? A. A is the answer. Right? A is the answer. Beautiful. Look at the next question now. Look at the next question. Why is it not going on question? Now, which of the following statements are correct about respiration? Respiration. Energy of oxidation reduction is utilized for photophosphorylation. Yes, you learned throughout this chapter. Oxidation reduction is happening. Energy is used in your phosphorylation to make ATP, adding of ATP and everything. In glycolysis, you learned that. Right? Respiration. Oxygen act as the final electron acceptor. Yes, we learned that. The photooxidation energy is utilized for production of proton gradient required for photophosphorylation. No. Why? Because photooxidation energy is under it is for photosynthesis. It is for photosynthesis, not for respiration. For respiration, we have what? Oxidation reduction energy. Remember that point. Wrong statement. The role of oxygen is limited to the terminal end of the respiration process. Yes. Proton cross the outer membrane of mitochondria through the channel formed an integral membrane protein complex. No. Why is it no? Because it is not outer membrane. It's supposed to be inner membrane. It has to be inner membrane. So what is the answer? A is correct, B is correct, and D is correct. ABD. Yes, ABD. 2 is the answer. 2 is the answer. Right? 2 is the answer. Right? Next question. Complex number 2. Tell me, this is a very easy question. This is a very easy question. You cannot go wrong on this. You take 2 minutes and do this. We have learned this already. What is complex number 1? NADH dehydrogenase. Complex number 2 is going to be your FADH2 dehydrogenase. Complex number 3 is going to be your cytochrome B7 complex. Then we have cytochrome AC3 copper synthase. That is complex number 4. Easy question. Next question. Identify the cytochrome which act as a mobile carrier for transfer of electrons between complex 3 and complex 4. That was your mobile carrier. That is going to be your cytochrome C. Again, NEET PYQ. Again, a NEET PYQ. Very easy. Chapter is very easy, students. Understand the concept and marks is yours. Next concept, Shri. Which of the following statements are incorrect? This question also is a NEET PYQ, year is not mentioned, is incorrect. ATP is synthesized through complex number 5, yes. Oxidation reduction reaction produces proton gradient in respiration, yes. During aerobic respiration, the role of oxygen led to terminal end, yes. The electron transport chain, one molecule of NADH gives 2 ATP, no. It has to be 3 ATP. One FADH gives 2 ATP. So this is a wrong statement. This is a wrong statement. Done. Now, that was the end of your electron transport chain. That was the end of your electron transport chain. Did all of you understand? Were you able to solve the questions based on what you learned in this class, students? Based on the concepts which you are learning today, you are able to solve the beautiful questions without any difficulty. Without any difficulty, right? Without any difficulty. Now, the next concept is very simple. It's going to be your call for backup. I always teach this in a very simple fashion, very simple to understand. Whenever your mother is not in your house, what do? You will make Maggie, stuff the stomach with Maggie, or you will order from Zomato or Swiggy. That is your backup. That is your backup. Similarly, inside your body, when there is no carbohydrates, inside your body there is no glucose, your body looks for a backup, and the backup is going to be fats and proteins. The backup is going to be fats and proteins are going to be your backup. That is breakdown of fats and breakdown of proteins. That is your backup inside your body. Backup inside your body. For example, for proteins, for fats, how do you break down the fats? So all of you should know there are two types of fats. One is called as your fatty acid, fatty acids. The other one is called as glycerol. Other one is called as your glycerols. Your fatty acids undergo something called as beta oxidation. Undergo something called as beta oxidation to produce your acetyl-CoA. To produce acetyl-CoA. Now tell me, this acetyl-CoA, can it enter the Krebs cycle? Yes. Enter the Krebs cycle. It enters the Krebs cycle. And if it enters the Krebs cycle, what will happen? NADH and FADH is produced. If FADH and NADH is produced, the FADH and NADH will enter the electron transport chain. ATP is produced. Sorted. Life is sorted. Fats are produced. Fats are able to produce energy. Simple. Now what about glycerol? When glycerols undergo oxidation, they produce something called as DHAP. They produce something called as DHAP. Now, what is the DHAP, students? Where can you use the DHAP? This DHAP can isomerize into GAP or PGAL, phosphoglyceraldehyde, right? Phosphoglyceraldehyde or glyceraldehyde, right? This can get converted to pyruvate. Pyruvate can get converted to acetyl-CoA. Acetyl-CoA will enter the Krebs cycle. Krebs cycle will produce energy with the help of electron transport chain. Done. DHAP, right? That is dihydroxyacetone phosphate. So this is called as alternative pathway. This is called as alternative pathway in which a particular fat or a protein can take an alternate pathway when there is no glucose in the body. When there is no glucose in the body. For example, similarly, in the case of proteins, transformation fat, this is acetyl-CoA. See, fatty acids can convert to beta oxidation, acetyl-CoA, FADH2 and NAD+ can enter the Krebs cycle. Similarly, which we learned right now, whatever we learned right now. Now, have some of you heard there is something called as burning of fats? Students, let me tell you very clearly, if you want to decrease your weight, if you want to decrease the weight, and the only way, the actual way is undergoing calorie deficit. The meaning of calorie deficit is that you take the less amount of calories and try to burn the fats. Unless the fats are completely consumed, your carbohydrates will not burn. Okay, so if you want to burn the fat, reduce the carbohydrates. Until there is no carbohydrates, fats will not burn. Fats will not burn at all. Because your body knows there is carbohydrates. Why should they use fat, right? Why should the body burn fat at all? right? So if you want to burn the fat, decrease the carbohydrates, fat will start to be utilized. The fat will start to be utilized. Okay, that's called burning of fats. Now, what about proteins? What about in the case of proteins? Proteins is very simple again. Proteins again is very simple. It can be, all of you know, proteins can be broken down to amino acids. Proteins can be broken into amino acids. For example, there is one amino acid called as glutamic acid. Glutamic acid, when it undergoes deamination, it will produce your alpha-ketoglutarate. Alpha-ketoglutarate is a part of your Krebs cycle. It will undergo Krebs cycle. Krebs cycle will go to undergo, later we have electron transport chain, then you have ATP. Very simple, life is sorted if you break down proteins. Similarly, we have something called as aspartic acid. Aspartic acid, when it undergoes deamination, it produces something called as oxaloacetic acid. It is the same oxaloacetic acid which can undergo Krebs cycle and produce then you undergo electron transport chain, produce ATP, right? Or produce ATP. Clear? Then, so did we learn till now? Whenever we have, when we come to your respiration, we always believe it as complex substances. Glucose is broken down to produce energy. That is called as catabolism. Complex substance such as your glucose is broken down to produce your simple substance such as ATP. But did we also learn just now that your proteins and fats can also break down to produce complex substances? Yes. Is it not anabolism? Yes, it is anabolism. So, remember very carefully, your respiration is going to be a combination of both catabolism and anabolism, and together we call it amphiboic pathway. Together we call it amphiboic pathway. Both both catabolism as well as anabolism is there. Didn't we just learn right now? Complex substances as your proteins are breaking down. Okay. This is your NCERT snip. See this? Very easy to understand. We have fats, carbohydrates and proteins. Proteins, amino acid, I told you. Aspartic acid and glutamic acid deamination can produce your acetyl-CoA. It can produce your alpha-ketoglutarate, right? Enter here. Similarly, your fat can enter your, directly become acetyl-CoA and enter the Krebs cycle. So, alternative pathways can be taken. For fats, it is usually acetyl-CoA. For fats, fatty acid is always acetyl-CoA. Remember that. See this question, very simple question again. It's a NEET PYQ, NEET 2023 question. Fatty acids are connected to respiratory other pathway through. Didn't we just learn fatty acid can get converted to acetyl-CoA? That is a simple answer. NEET PYQ. Right. Now, last concept for today is going to be respiratory quotient. Last concept for today. Respiratory quotient. It tells you that, very simple. Volume of CO2 evolved by the organism divided by the volume of oxygen consumed by the organism. Now, this can vary based on your substance, which substance are you using. For example, if there is for glucose, equal amount of oxygen, equal amount of CO2. Both are equal. So, respiratory quotient is going to be 1. It's going to be 1. What about in the case of your fats? In the case of fats, it's not very same obviously. In the case of fats, we have more CO2. So, we have less CO2 consumed, more oxygen being consumed. Can you see? They are taking more oxygen to break down the fat. More oxygen is required to break down the fat. Example of fat is given here. Example of fat is given here. That is going to be your tripalmitin. Is a fat example. So, respiratory quotient is around 0.7. 0.7 for fats. Similarly, for protein also, again, we require more oxygen, less CO2. The respiratory quotient is around 0.9 for proteins. And we have a question, last question. So, before we go, same thing is given here. This is NCERT snip. See here, 0.7 for your fats, 1 for carbohydrates, 0.9 for proteins. This is your homework question. Let me know in the comment section what is the answer for this. The respiratory quotient value for tripalmitin, which is a type of fat, is what is the answer? This is a NEET 2019 PYQ. NEET 2019 PYQ. Now students, Didn't we just do so many questions? Yes, we did so many questions. Now the thing is, you understood the chapter. You were able to solve the question. Some questions you are not able to solve. Now you will obviously need to solve more questions. You need a proper test series, right? Obviously you need a proper test series, right? You need a test series. So if you go to the description of today's video, you will find a link, students. You will find a link that is very important right now. It's not right. The thing is, I've always told to all the students that writing test paper is actually the key. If you start from now only writing enough of mock tests and everything, you will understand the concept even more beautifully and you'll be able to solve more questions. So if you go to the description right now, here, there's something called as test series. 1 year long test series. It's not for 1 day, 2 day, 3 day or few months. It is a year long test series, students. Year long test series. See here. So we have totally 25 test series. Total 25 tests. 7 full mock tests, 12 part tests and 6 subject wise tests. 6 subject wise tests. So if you don't want to waste any time and start the test series right now without wasting any time, this is the chance where you go. You can buy the test series right here. It's for just 1,000 rupees. It's reduced. The price is reduced. 15 to 25 for books also. Okay, it's a book also. You'll be getting a book. Okay, so you just for 1,000 rupees you have 25 tests. 1,000 rupees, 25 tests. Everything is given here. You can buy it directly from here or add to the cart. I think add to the cart. I'll just keep it here for myself. Okay, so students, that is the entire chapter of your respiration plants. Let me know in the comment section how was the class. And if you did not understand any part, tell me in the chat. If you understood some part very clearly, that also tell me in the chat. Okay. And the next chapter we'll be doing and finishing the entire plant physiology is going to be your plant growth and regulators. And I'll see you in the next class. Take care. Bye-bye. And have an amazing day and night. Bye-bye, students.